ಮುದ್ದಿನ ಮಕ್ಕಳೇ, ಹೆಂಗಿದ್ದೀರಾ? ಚೆನ್ನಾಗಿದ್ದೀರಾ? Students, today I, Dr. Smita Ma'am, is going to discuss the NEET 2023 paper. Yes, students. ಹೆಂಗಿತ್ತು paper, students? Was nice or very nice? Yes, students. If you analyze the paper was actually very easy. Technical problem, if you see, the paper was actually very lengthy because ತುಂಬ assertion reason ಮತ್ತು statement based questions ಎಲ್ಲ ಇತ್ತು. So definitely because of that paper ತುಂಬ lengthy ಇತ್ತು, but otherwise questions were very easy. If you see, the straightforward questions were directly picked from word to word, that is from the NCERT. As I used to say, you please refer to NCERT text line to line, word to word. Yes, students. Let us now discuss the paper. So Smita Ma'am from Diksha, Karnataka is now going to discuss this paper in front of you. And yes, students, let us see how much you can score, how many marks and where all you have lost your marks. Okay. Come to the first question. And here we go. The first question, that is the first question in biology 2023 paper. Given two statements, one is a reason and assertion, right? Assertion. The first stage, okay, of gametophyte, okay, in the life cycle of moss, that is the protonema stage. If you recollect, protonema stage is the initial stage, okay, which later gives rise to something called as prothallus, a very, very important structure of the life cycle. That is the prothallus. This prothallus later, if you see, ಅದರಲ್ಲಿ ತುಂಬ two main structures ಇರುತ್ತೆ. That is nothing but the antheridium and the archegonium. Yes, students, this is a very important part that is called as the prothallus. Again, a very true statement, that is assertion correct ಇದೆ. ಆಮೇಲೆ reason ಏನಂದ್ರೆ protonema develops directly from the spores produced in the capsule. If you see, assertion, reason are both correct. And yes, moreover, option A is not correct but R is correct. Second option, both A and R are correct, and R is a correct expression. So the answer here is option second, right? Okay. Next question. Cellulose. Cellulose does not form blue color with iodine. You know, it's a very detection test, very common test done. But if you see, when we use starch, we get the blue color. Why? Because of the structure of the starch, which is able to hold the iodine. But here in the case of cellulose, if you see, you cannot form the blue, you cannot get the blue color with iodine, and that is because it does not contain complex helices, and yes, cannot hold the iodine molecule. So answer here is the fourth option. Next one, which micronutrient require for the splitting of the water molecule? That is a very important step considered to be in the photosynthesis, and that is because of this manganese micronutrient, that is a small nutrient required in the lesser quantities. Expressed, next question, express sequence tags, okay, that is ETS, refers to certain important expressed genes. No. All genes that are expressed as RNA, or the third is all genes that are expressed as proteins, all genes whether expressed or unexpressed. If you see, again, this is not the answer here. Let us see about these two now. If you recollect the central dogma principle of molecular biology, central dogma andre ಏನು students? We studied in the chapter molecular base of inheritance, right? There what we had studied, you know, the DNA. The very important DNA has got all the information present within itself, and yes, adu duty DNA ಏನು ಮಾಡಬೇಕು ಅಂದರೆ express ಮಾಡಬೇಕು informationಗೆ, and expression of the information happens in the form of the proteins. And you know very well, as per the central dogma principle of molecular biology, DNA forms the mRNA, right? mRNA, and that was nothing but the transcription process. We had studied that. This mRNA undergoes then process called translation. Andre ಏನಾಗುತ್ತೆ, it will express its information, yes, and that is in the form of the protein. So if you see, our option B is a correct answer here, that is expressed as the, all the genes expressed as the RNA, right? Next one, the thickness of the ozone layer. ಏನು ಇದು ಓಝೋನ್ ಲೇಯರ್ ಫಂಕ್ಷನ್ ಸ್ಟೂಡೆಂಟ್ಸ್ ಹೇಳ್ತೀರಾ ಸ್ವಲ್ಪ? ಎಸ್, ಓಝೋನ್ ಲೇಯರ್ ಆ್ಯಕ್ಚುಲಿ ಫಿಲ್ಟರ್ಸ್ ದ UV ರೇಡಿಯೇಷನ್, ಆ್ಯಂಡ್ ಎಸ್, ಇಟ್ ಡಸಂಟ್ ಅಲಾವ್ ಅಸ್ ಇಟ್ ಟು ಫಾಲ್ ಆನ್ ಅವರ್ ಸ್ಕಿನ್. ಇಫ್ ಯು ರಿಕಲೆಕ್ಟ್, ದ skin isn't protected undergoing with further, if the UV falls on the skin, it causes a mutation, the damages, it can cause skin cancer also. But yes, we should thank this ozone layer, because of that we are protected from the harmful UV radiation. And yes, it is nothing but measured in terms of Dobson units. A very, very straightforward question, student. Given below are two statements, okay? One is labeled as assertion and the other is labeled as reason. Okay, let us now try to understand what is assertion. ATP is used at a two-step in glycolysis. It's a very important process, glycolysis, right? First, that is reason, first ATP, okay, is used in converting glucose into glucose 6-phosphate, and the second ATP is used in conversion of the fructose 6-phosphate into fructose 1,6-diphosphate. Completely true. If you see, both of them are true, provided also the reason is an explanation for the assertion. So let us try to find out the option here. A is true, but A is false, R is true, no. A and R are true, and yes, R is a correct expression for A. So this is option second here, right? Upon exposure to UV radiation, the DNA stained with iridium bromide gives you again a very straightforward question, that is a bright orange color. Next question, among the aryl quadrate, okay, which one, okay, is considered to be the most significant or important cause driving the extinction of the species? Extinction of the species, very important reason ಏನಂದ್ರೆ, let us see, is it co-extinction or habitat loss and fragmentation or the overexploitation or alien species? Of course, these all, amongst all the four, the very critical factor is actually found to be habitat loss. Yes, student, habitat loss and fragmentation is found to be the very significant step, or you can say problem, which is actually leading to extinction of the species. Which of the following stages of meiosis involves division of centromere? I hope you remember cell division we had studied mitosis and meiosis. In that meiosis, we had studied meiosis 1 and meiosis 2. Out of that meiosis 1 and 2, we need to understand where is the division of centromere happening. If you recollect the structure of the chromosome, right? You know, the sister chromatids are separated at the centromere, and that happens at what stage? That is anaphase 2, right? Again, it's a very important step in meiosis. Question 10. Which hormone promotes internode or petiole elongation in deep water rice? And it is found to be actually the ethylene which causes this effect. Next one, okay. Frequency of recombination between gene pairs, okay, on the same chromosome as a measure of the distance between genes to map their position on chromosome was used for the first time by Thomas Morgan's student, option 4, Alfred Stuyvesant, and he was the one who actually conducted this work, okay. He was student of Thomas Morgan. Next question, again a very important question. How many ATP and NADPH2 are required for synthesis of one glucose during the one molecule of glucose during Kelvin cycle? And right, the answer here is 18 ATP and 12 NADPH2. Next one, RNA polymerase 3. Remember RNA polymerase 3? Even ಮಾಡುತ್ತೆ transcription, right? And it helps for the synthesis of RMT, that is transcription of tRNA, snRNA and srRNA, right? The three types of the RNA. Next question, a very, very important question with respect to the morphology of the stamens, right? Family Fabaceae, okay, differs from Solanaceae and Liliaceae. How? With respect to stamens, okay, stamens, pick out the characteristics specific to family Fabaceae but not found in Solanaceae or Liliaceae. If you see, the very important characteristic shown by the Fabaceae is a 9+2 arrangement, 9+2 arrangement of the stamens, right? That is a very, very specific feature shown by the Fabaceae but not in the case of the Solanaceae. And yes, Solanaceae, if you see, it has shown the dithecous anthers, right? So this was a very important feature, especially you should always remember the diadelphous condition 9+2 condition of the stamen arrangement. Next one, the process, okay, which process is that? The process of appearance of recombination. Recombination andre first of all strike agbeku, it is meiosis 1, and in that meiosis 1 if you see there is a prophase 1 which undergoes the Packettine stage. Yes, students, it is the Packettine stage. Therein, if you remember the chiasmata formation, the Packettine stage, I am talking about that, and there is the formation of the recombination nodules at the stage that is prophase 1, and the answer is Packettine. And if you remember, it was that Packettine, actually because of the gracefulness of that Packettine, we are not completely looking like our parents. Yes, students, the difference in our parents and us lies, the slightest difference which occurs from us to them is just because of the Packettine stage taking place. Imagine if there wouldn't have been Packettine taking place, we all would have been looking like the Xerox copies of our parents. And that is the beauty of the Packettine, which actually allows us to remain little different from our parents, though genetically correct, but slight changes would be there. Okay, the next question. In the equation GPP minus R NPP, what is the GPP? Gross primary productivity. NPP is a net primary productivity. So what is R here? Okay, let us talk about that now. Reproductive allocation, no. Photosynthetically active radiation, again no. Respiratory quotient, again no. And yes, that is the respiratory loss. Next one, very simple, and ನಾನು ಹೇಳಿದೆಲ್ಲ paper ತುಂಬಾ easy ಇತ್ತು. These are very straightforward questions, students. Reaction center PS2 ಅಂದ್ರೆ directly ನಿಮಗೆ ಗೊತ್ತಾಗ್ತದೆ, that is 680 nm, right? A very straightforward question again. The next one, unequivocal, right? Proof that DNA is a genetic material. I hope remember there were many experiments like Griffith's experiment and the Hershey Chase, etc. were performed, and there was one experiment that was a Hershey and Chase experiment which was actually talking about the bacteriophage use ಮಾಡಿದ್ದು, bacteriophage ಆಮೇಲೆ ಅದು infect ಮಾಡಲಿಕ್ಕೆ use ಮಾಡಿದ್ದು E. coli ನಲ್ಲಿ, and then he had used radioactive phosphorus and radioactive sulfur, and that Hershey and Chase experiment proved that DNA is a genetic material but not the protein. And the answer here is Alfred Hershey and Martha Chase, a very famous experiment done to prove that DNA is a genetic material. Spraying of which of the following phytohormone on juvenile, young conifer helps in hastening the maturity period, and that is found to be GA, gibberellic acid, which does the influence of the production of the early seed production. Next question, what is the function of the tassels in corn cob? Tassels andre ಏನು student? Imagine now this is a corn cob and these are the tassels. So what is this tassels going to do? Don't you think, looking at the structure, it acts like trying to trap the pollen grain? Yes, that is a case which is found in the case of the wind pollination, wherein you can see it will help trapping the pollen grains and will help for the pollination purpose like that. So that is a very important function of the tassels in the corn cob. Next question. During the purification process of our DNA technology, chilled ethanol will precipitate out. Again, very straightforward answer, and that is the DNA. ಇದು ನೀವು DNA recombinant technology process ನಲ್ಲಿ ಇದು one of the very important steps study ಮಾಡಿದ್ದೀವಲ್ಲ, that is pooling technique, right, which helps for the extraction, or you can say precipitation of the DNA. In angiosperm, in angiosperm, the haploid, diploid, triploid, structures of a fertilized embryo sac. When I talk about the fertilized embryo sac, try numbering them. Synergids N, again no, N, no, N, no, N, 2N, 3N, right? And the answer here is option 4. Isn't it a very simple question, student? If you know the structure, then things are very simple here. Large, when I talk about large, colorful, fragrant flowers. Large, colorful, fragrant flowers, of course, will not be needed by them. But yes, very importantly needed by these creatures for insect pollination plant. That is pollination taking place with the help of the insect, right? Okay, next one. In tissue culture experiments, leaf mesophyll cells andre the meristem, which is a permanent tissue, and from that permanent tissue we are putting that permanent tissue, imagine now this is a nutrient medium and we are putting that permanent tissue mesophyll to initiate a callus. Now don't you think from the mesophyll, and mesophyll andre ಏನು permanent tissue, right, meristematic tissue, and from the meristematic tissue actually mesophyll cells we are trying to form the callus. Andre don't you think we are actually trying to form the callus, which is nothing but from the meristematic tissue. Since it is the process forming the callus, we are actually going back, that is differentiation, right? Again, it's a very tricky question. From the leaf mesophyll cells we are trying to form a callus, and that is nothing but differentiation. Given below are the two statements. The forces generated by transpiration can lift xylem sized of column of water 130 meters height. What is this? This is the first statement. If you recollect, in transport in plants chapter nanali nodidivi alla, what is the main reason for conduction of water against the gravity towards the tip of the plant? ತುಂಬಾ ಥಿಯರೀಸ್ ನಾನು ಅಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಿದ್ದು. ಒನ್ ವಾಸ್ ದ ರೂಟ್ ಪ್ರೆಷರ್ ಥಿಯರಿ. ರೂಟ್ ಪ್ರೆಷರ್ ಥಿಯರಿ ಆ್ಯಕ್ಚುಲಿ ಸ್ವಲ್ಪ ಫೇಲ್ ಆಗುತ್ತೆ. ಯಾಕೆಂದರೆ ರೂಟ್ ಪ್ರೆಷರ್ ವಿಚ್ ಇಸ್ ಕ್ರಿಯೇಟೆಡ್, ಅದು ತುಂಬ ವೆರಿ ಲೋ ಲೆವೆಲ್ ಆಫ್ ದ ರೂಟ್ ಪ್ರೆಷರ್ ಇಸ್ ಕ್ರಿಯೇಟೆಡ್, ವಿಚ್ ಇಸ್ ಹಾರ್ಡ್ಲಿ 1 to 2 atmosphere pressure, which is not sufficient to conduct water for a 100 feet tall plant. Root pressure theory definitely valid ಇದೆ, but for which type of plant? Small sized plants ಇರುತ್ತಲ್ಲ, herbs or shrubs. So ಅದಕ್ಕೆ root pressure theory work ಆಗ್ಬೋದು. But when we talk about the 100 feet tall plant like the coniferous plant, root pressure theory work ಆಗುದಿಲ್ಲ. So that's a reason there were many theories explained, and then there was a very important theory, that was the transpiration pull theory, which said that it was a transpiration pull which helped for the conduction of water for a 100 feet tall plant also. Yes, student, this transpiration pull theory is able to give water for all the tall heighted plants, and yes, the statement 1 is very correct. Let us see the statement 2. Transpiration cools the leaf surface sometimes 10 to 15 degrees by evaporative cooling. Again. This is also very true, students. ಯಾವತ್ತು transpiration ಆಗುತ್ತೆ, definitely ಆ ಟೈಮ್ಗೆ ಏನಾಗುತ್ತೆ, because of the too many reactions happening here, the heat is generated, and literally you should say leaves are saved from that high temperature. Because of that evaporation, actually the temperature is been maintained. The temperature is maintained and it gives a cooling effect. Again, a very important attribute shown by the transpiration pull theory, followed by the process of the evaporation also. So if you see both the statements are true here, let us go to the options here. Statement 1 is incorrect, but 2 is correct. No, both statement 1 and 2 are correct. Option 2 is the answer here. The next question. The historic convention, the historic convention on biological diversity Earth Summit was held in Rio de Janeiro, and that was in 1992. Again, a very important question, and a repeated question, and a very straightforward too. Yes, very famous question. In gene gun method, gene gun, ballistic or gene gun method, which is used to actually introduce the whole, or you can say the foreign DNA, right? Foreign DNA into the host cells. Which particles are we going to use? Yes, very simple. Why are we using this? Because it is not reactive. Yes, that's the reason ನಾವು ಏನು ಮಾಡ್ತೀವಿ, tungsten or gold particles use ಮಾಡ್ತೀವಿ, and the answer here is tungsten or gold because it is not reactive, right? Okay, next question. Movement, movement and accumulation of ions. When we talk about accumulation of ions across a membrane against their concentration gradient. Whenever we talk about against the concentration gradient, you should understand that yes, the answer is going to be active. Against the concentration gradient, that is active. That is very important aspect. Again, we have studied that in transport and plant, students. Next question. Exile placentation. I hope you remember exile placentation andre ಏನು students? There was some kind of placentation wherein you can see this kind of arrangement. Sorry, I repeat that once again. Kind of wherein you can see such kind of the arrangement, right? Axile placentation. And yes, this was shown in the case of what? Very simple. China rose, petunia and the lemon. This combination of the plants will show you the axile placentation. Next question. The next question here is, identify the correct statements. Detrivores perform fragmentation. Yes. The humus is further degraded by some microbes during the mineralization. Yes. Water-soluble inorganic nutrients go down into the soil and get precipitated by process called as leaching. Again, yes. Detritus food chain begins with living organism. No, it begins with dead organism, right? And earthworms break down detritus into smaller particles by process called catabolism. Sorry, it is wrong. It is fragmentation. So the answer here, let us look at the options here. Correct answer from the option given below, either DEA or ABC. Let me go back again. It was ABC. So the answer here is going to ABC, right? Okay. Next one. Among eukaryotes, okay, among eukaryotes, replication of DNA, a very, very simple question, students. Look at the straightforwardness here. If you remember G1, S, G2 and the M phase, the cell cycle, right? And where is the replication going to take place? Yes, that's going to take place in the S phase, that is the synthesis phase. Look at the question, students. It is so simple actually. Given below are two statements, endarch and exarch. Okay, endarch and exarch are the terms often used for describing the position of secondary xylem in the plant body, which is actually not correct. Not correct. And what about the next one? Exarch condition is the most common feature of the root system. Yes, that is again a correct option. Correct statement. Let us see which one is the correct one here. Statement 1 is incorrect, but statement 2 is true. Statement both 1 and 2 are true, both 1 and 2 are false, 1 correct, 2 wrong. Actually, statement 1 is incorrect and 2 is correct. Option 1. The phenomena of pleiotropism. What is pleiotropism? Single gene, okay, single gene showing multiple phenotypic expression, multiple effects. Yes. So what is the answer here? More than 2 genes? No. Presence of several alleles of a single gene controlling a single crossover? Again, no. Presence of 2 alleles? Again, no. Each of the 2 genes controlling a single trait? No. Yes, a single gene affecting multiple phenotypic expression, and that is nothing but here the case, that is pleiotropism, right? Okay, next one. Identify the pair of heterosporous pteridophytes. Heterosporous under ಏನು students? Different. Okay, different spores been produced. Homosporous under same. Heterosporous under different. Very simple. Equisetum and Salvinia, Lycopodium, Selaginella. Yes, Selaginella and Salvinia, very important repeated question. Selaginella and Salvinia is the answer here, which shows the production of the 2 different type of the spores in the case of the, that is the pteridophytes. Assertion reason question. Late wood has fewer xylary elements with narrow vessels, and cambium is less active in winters. Okay, if you recollect, very true. Both are very true, students. If you see, cambium actually yavaga active irutte during the spring season. Very true, it is inactive during the winter season, right? So let us look at the options here now. A is false, but R is true. No, both A and R are true, and R, yes, is a correct explanation of A. Very correct, student. In that, out of the 15 questions, candidates can choose to attempt any 10 questions. Really, question note was, students, identify the correct statement. Lenticels, okay, lenticels are lens-shaped openings permitting the exchange of gases. Yes, it is again true, right? It's a true statement. Okay, true. Bark formed early in the season is called as hard bark. Again, true. Bark is a technical term that refers to all the tissue exterior to vascular cambium. False. Bark refers to periderm and secondary phloem. Yes, that is again true, right? Okay. Phylogen is a single layered in thickness. Oh, that is false. Okay, if you go to this option, let us check which is the correct option here. Correct answer for the option, we found that option A, B and D were correct. Let us search this option. Yes, and it is option 4th here, which is found to be the correct answer. Match list 1 with list 2. We come with the very simplest one. Quiescent stage is nothing but the inactive phase, right? I'm talking about the cell cycle, okay? G1, S, G2 and the M phase. M phase is the equational division, right? G1 is going to be the interval between the mitosis and initiation of DNA replication, and G2 is synthesis of proteins, right? So we have paired A4B1C2. A4B1C2, and yes, that's the correct answer here. That is option C. That is the option what I said here. M phase with equational, G2 with protein synthesis, quiescent is nothing but inactive, G1 with the interval between the initiation or the mitosis. Okay, the next question here. Given below are the two statements, okay? One is assertion and second one is a reason, okay? In gymnosperms, right, the pollen grains are released from the microsporangium and carried by the air currents. Oh, again it is correct, right, true. Air currents carry the pollen grains to the mouth of the archegonia, where the male gametes are discharged and pollen tube is not formed. Oh, I'm very sorry to say, students, it is not directional movement of the pollen grain, so they cannot be specific talking about the mouth of the archegonia. It is not the correct one, not correct, right? So let us now go to the option here, right? A is false, but R is true. Both A and R are true. Sorry, it is not the answer here. Both A and R are true, but R is not the correct explanation. A and R are true, they are not true together. A is true, but R is false. Yes, students. So A is true and R is false. Correct. So option 4th here, right? Okay. Again, match list, that is list 1 with 2. If you see, the very easiest to remember, zinc with synthesis of oxygen, Zn, right? Iron with activator of catalase, right? Boron helps for elongation and differentiation, and molybdenum with component of nitrate reductase. Now let us match and correct with the option here. A with 3, A3B1C4, A3B1, A3B1C4. Yes, the answer here is option 4th. Which of the following combination is required for chemiosmosis or chemiosmosis, right? So let us, chemiosmosis is nothing but the movement of the ions, the proton movement, right? Again, on the membrane. If you see, what are the things required? Look at this beautiful image. It will help you to understand everything. We need a membrane first of all, right? Correct, right? Secondly, we also need the proton pump, right? We need the ATP synthase also. So look at the options here. We need the proton pump. We need the membrane, correct? Proton pump, very good. Of course, very important, a proton gradient is required. The difference, okay, in the proton gradient is required. And yes, ATP synthesis. So answer is option 2. So the next question here, main steps, okay, in the formation of rDNA technology are given below. Arrange in the correct sequence. Look at that, it's a very simple question again. Insertion, no. First is going to be cutting of the DNA. Cutting of the DNA at specific location by REN, yes. Isolation of the desired DNA fragment, amplification of gene of interest using PCR, okay. This is going to be the second one, right? And the third one, amplification, and the fourth one, okay. Let us now go back to the options here. So this is going to be B, C, D and A. Okay, let us search this option here, and the option here is second, B, C, D, A. The next question, okay. Which one of the following is wrong, not correct? The amount of some toxic substance of industrial waste water increases in the organism at successive trophic level. Yes, very, very true. It is actually correct, right? Yeah, that is what you can say, the bioaccumulation, what we say, right? Pollutants getting into the food chain. Next one, the microorganism involved in the biodegradation of organic matter in the sewage polluted water body consume a lot of oxygen, causing the death of the organism. Again, true. Algal blooms caused by excess of organic matter in water improve the water quality and promote fisheries. Sorry. It will not improve. In fact, it will deteriorate. It will deteriorate the water content, and it will lead to the death of the life present inside the water body, because it will trap the algal bloom, actually will trap and ಅದು oxygen CO2 supply ಮಾಡಲಿಕ್ಕೆ ಆಗುದಿಲ್ಲ. That's the reason there is no, there is deterioration of the life beneath the algal layer. Okay, so the answer here is going to be the option 3. Before that, let us see again. Water hyacinth grows, yes, very correctly, abundantly in eutrophic. You know what is eutrophication? Excess nutrient being flowing onto the water body and enriching the life of the organism there to a very higher extent. Okay, eutrophism, water bodies leads to imbalance in the ecosystem. Yes, again this is true. So the answer here is going to be option C. Next one, okay. Which of the following statements are correct about Klinefelter syndrome? Syndrome was discovered by Langdon Down. No. Such an individual has overall masculine development. Yes, that's a very important feature shown. So very true, right? Affected individual is a short stature. No. Physical, psychomotor and mental development again retarded. No. Such individuals, yes, they are sterile. So according to this option, the statement correct is B and E. Okay, the option 4th is the correct answer. Statement B and D is found to be the correct answer. Okay, very simple. Look at that question. Noಡ್ತೀರಾ ಎಲ್ಲ simple, straightforward. Yes, I said lengthy question. Now this all question takes more time. Okay, mutualism and the equal help. Okay, no damage occurring, so you should search for plus. So mutualism is going to be the plus here, right? Commensalism plus 0 ಎಲ್ಲಿದೆ, +0, right? Amensalism -0 ಇಲ್ಲಿದೆ. Okay, parasitism plus and minus. So where is the plus and the minus? Okay, be very careful. Okay, A4 B1 C2, A4, okay, B1 C2, B2, A4 B1 C2. Okay, let me go back again. C2 and D3. Yes, so option correct here is the C, that is the third option, right? Okay, the next question here. A flower is defined as a modified shoot wherein the shoot apical meristem changes to floral meristem. Again, yes, it is correct. Internode of the shoot gets condensed to produce different floral appendages laterally at successive nodes instead of leaf. No, it is not correct. Okay, let us go to the option here. A is false. No, A is true. In fact, B is false. A and R are true and R is the correct. A and R are true. A is true but B is false. The answer here is option 4th. How many different proteins does a ribosome consist of? That is again very simple, 80. Okay, 80 proteins. Again, match. Okay, cohesion. I hope you remember the transpiration pull theory. So this match is related to the transpiration pull theory. Cohesion, attraction between the similar molecule. Adhesion, dissimilar molecule. So cohesion, try to match with the similar molecule, right? Mutual attraction amongst water molecule. Cohesion. Adhesion is going to be attraction of towards the polar. Okay, so the answer here with this. Surface tension, guttation. Guttation is very simple to understand. Guttation is water lost in the form of liquid, hydatodes, guttation. And then surface tension, more attraction in the liquid phase. Okay, now let us try water loss. Okay, let us try to connect this with the option A2B4C1. A2, okay, A2B4 C1 and D3 A2, this should be 2. Okay, by mistake it has come as I, it should be B2, sorry, A2. Okay, Roman letter 2, the option here A2, B4, C1. B4, C1 and D3. Yes, so answer here is option 2, right? Okay. Again match the question here, options here, okay. So very simple. When you talk about the TCA, citric tricarboxylic acid cycle, go with citrate synthesis. Glycolysis, that is the EMP pathway. Oxidative phosphorylation, electron transport, and oxidative decarbonylation, that is pyruvate dehydrogenase. Again, let us connect A to B4, A to B4, A to B4, okay. C3, A to B4, A to B4, C3, D1. The answer here is option 1. Malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of, what is it? Dinitrogenase, succinic dehydrogenase, amylase and lipase. That is succinic dehydrogenase. And next question. Statement 1. Gauss competitive exclusion principle states that two closely related species competing for the same resources cannot coexist indefinitely and competitively. Inferior one will be eliminated eventually. Correct. Very correct. And the statement 2 here is, in general, carnivores are more adversely affected by competition than herbivores. No, in fact, it is reverse. They are the affected ones. Okay, let us look at the correct option here. Option both 1 and 1 is incorrect. Second option, both statement 1 and 2 are true. Both statement 1 and 2 are wrong. Okay, these are not the correct here. Okay, left out is which one? Statement 1 is correct, 2 is false. Answer is option 4. Okay, so here students, we have completed with all the 50 questions. Yes, students. So here we completed the session from, in this video we completed the question from 1 to, that is from 101 to 150. And yes, students, in the next we will be continuing with question number that is from 51 to 100. So here students, we just sign off for the lecture today, and we'll be right back with our next session. Bye then students. Thank you.